This short lecture will be to review the metabolism of the major nutrients, carbohydrates, lipids, and proteins. This is continuing after our discussion of cellular or aerobic respiration, where you're taking a glucose molecule and breaking it down to obtain a large amount of ATP, which is it's known as the energy currency of the body. It's really the only molecule that can be used directly by body cells for energy. And now we'll just look at carbohydrates, lipids, and proteins individually to look at how they get broken down and also created by the body. So remember that catabolism is same thing as catabolic reactions. These are breakdown reactions where you take a larger molecule and break it apart into its smaller components, smaller molecules or atoms. And anabolic reactions are the opposite, where you take smaller atoms or molecules and put them together. Usually you're creating bonds between them to create a new, larger molecule. So we'll start with carbohydrates. Starting with carbohydrate catabolism, and this is performed through cellular respiration, which we have already discussed. Remember, cellular respiration involves 4 main steps, starting with glycolysis, ending with the electron transport chain. This shows you the overall reaction where you take glucose, you start with 1 molecule of glucose. It does require oxygen. And end products of cellular respiration are, first of all, a lot of ATP, and then carbon dioxide and water molecules. Carbohydrate anabolism. There's several different types of carbohydrate anabolism to create different types of carbohydrates. First of all, glycogenesis is when you take several glucose molecules and you put them together to create a larger glycogen molecule. So this is actually how glucose is stored when it's not needed immediately for energy. You'll take many glucose molecules and link them all together to create glycogen. If glucose is not used immediately for ATP, then it is stored as glycogen. This process, called glycogenesis, is promoted by insulin. So with these terms, you can look at the ending, and genesis means formation or creation. Lysis means breaking down or breaking apart. So the ending of these terms, you're going to see several of them coming up. It will tell you whether you're creating this molecule or breaking it down. And the first part of the word is going to tell you what molecule you are creating or breaking apart. So glyco in glycogenesis refers to glycogen. When you see gluco, that refers to glucose. So glycogenesis is the formation of glycogen. That's like the exact translation of that word. Genesis is creation or formation, and glyco refers to glycogen. All right, so moving on to the next one, glycogenolysis. This is the opposite, when you take glycogen and you break it apart into the individual glucose molecules that form the glycogen. So this will happen when glucose is needed for ATP. And this process is promoted by 2 different hormones, glucagon and also epinephrine. And the third term in your outline is gluconeogenesis. This is the formation of glucose from something that is not a carbohydrate. So you're not just making glucose out of glycogen. You're forming it from like a protein, for example. So this is going to occur in a couple different situations. If there's no more glycogen available to break apart and get glucose from, and that typically happens on a small scale every day. When you're in between meals, you haven't eaten in a while, your body's going to use its stores of glycogen to get glucose in order to get energy for your cells. This process will occur on a larger scale If an individual is starving or is eating very few carbohydrates, then they're going to have to turn to a different source, a different type of molecule for energy. And this process is promoted by the hormone cortisol. So the different substances that your body can make glucose from, there's really two main choices, either amino acids, which can be turned into pyruvic acid, and then pyruvic acid can enter glycolysis and be converted into glucose, or glycerol. And glycerol is one of the components of lipids, most lipids. Glycerol can be converted into glyceraldehyde 3-phosphate, which is a part of the process of glycolysis, and you can just convert that right into glucose. So using either one of those molecules, you can make glucose. All right, next is lipid metabolism. So basically, lipids have almost twice the amount of energy that carbohydrates do, but they're not used first because they're harder to break down. Glucose is the easiest thing to break down, so that's always the first source of energy. But lipids are a really good source of energy too. It just takes a little bit longer, a longer process to break them down. Lipids are stored in adipose tissue. And we'll start by talking about lipid catabolism, which is called lipolysis. So first of all, when you ingest lipids, and remember the most common type of lipid that you are eating in your diet are triglycerides. Triglycerides are broken apart by the enzymes in your digestive tract, by lipases, and also with the help of bile. They're broken into their two main components, which are glycerol and fatty acids. The hormones that promote that are cortisol, epinephrine, and norepinephrine. So then from there, though, what happens next? You have glycerol and fatty acids. Now you have to keep breaking those down. Those are still big molecules. You have to break down even further if you want to get energy from them. So for glycerol conversion, it is converted into glyceraldehyde 3-phosphate. That is part of the glycolysis pathway. And then your body will kind of assess what its needs are. If the individual does not need any more ATP at that moment, they have plenty of energy for their cells, then glyceraldehyde 3-phosphate will be converted into glucose and then stored as glycogen for later, whenever your body needs it. But if the individual does need ATP right away, then glyceraldehyde 3-phosphate will go through the glycolysis pathway and the rest of cellular respiration in order to get ATP, since that is needed right away. So that's what happens to the glycerol. It'll just go through glycolysis if energy is needed, go through glycolysis, go through the Krebs cycle, go through the electron transport chain to make a lot of ATP. For fatty acids, the way that fatty acids are broken down is through beta oxidation. This is when the fatty acids are broken apart into small pieces. Each of those pieces consists of 2 carbon atoms. These 2 carbon atoms, let me go back to this. These 2 carbon atoms are attached to coenzyme A. Remember, that's just a carrier molecule. And then that forms acetyl coenzyme A, which you can see here. So after beta oxidation, you just add coenzyme A to get acetyl coenzyme A. And that, if you remember, that is what enters the Krebs cycle. And then that can just be broken down through the Krebs cycle, through the electron transport chain to get ATP. The next term here in your outline is ketogenesis. So ketogenesis is when your liver cells actually take 2 of those acetyl coenzyme A molecules and combine them. And it'll do that if you have a whole lot of those molecules just hanging around in your blood. So it'll start taking them and putting them together. And when it puts them together, that forms acetoacetic acid. So this only happens if you have a whole lot of acetyl coenzyme A molecules kind of accumulating in the blood. So your liver makes acetoacetic acid. Some of that acetoacetic acid is converted into beta hydroxybutyric acid and acetone. And these three acids are written in your study outline. These three substances are known as ketone bodies. So these ketone bodies are now just in the bloodstream, and they can be used by other cells to make acetyl coenzyme A again, and just you can use them to make ATP. But it could lead to a condition called ketosis. That's when ketone bodies start accumulating in the blood. And that's often a result of starvation or a very, very low carb diet, or if you have diabetes and it's untreated. And this can be, it can be dangerous because it could lead to metabolic acidosis because these ketone bodies are all acids. So if they're building up in the blood, it can make the pH of your blood go down below the level where it should be. Okay, so next is lipid anabolism, also called lipogenesis. And this will occur just basically when more calories are ingested than are actually needed to make ATP. So if you ingest excess triglycerides, they just get stored as they are. If you ingest excess amino acids or excess glucose, they can be converted into triglycerides and then stored as fat, as triglycerides. And excuse me, triglycerides are stored in adipose tissue. That's the fatty tissue in your body until they're needed to make ATP. So adipose tissue stores about 98% of all your energy reserves. The other 2% is glycogen. Okay, so now protein metabolism. So, excuse me, no matter what, proteins are broken down into amino acids. But from there, they can be either used for energy or used to make new proteins or stored for later. So we'll start with protein catabolism. Like I said, proteins are always broken down into their individual components, which are the amino acids. And this is promoted by the hormone cortisol. And typically, though, after proteins are broken apart, those amino acids are reused to just build new proteins. Proteins do so many different things in the body. There's so many different types that have so many important functions that typically your body does have a need for a new protein. So it will just use these amino acids and reconfigure them to build a new protein, whatever is needed at the time. However, if amino acids aren't reused to make new proteins, then they can be broken down and used for energy. So amino acid breakdown involves just a couple steps. First, transamination, which is when an amino acid transfers its amine group to a different acid, and that forms a keto acid. Then that's followed by deamination, which is the removal of the amine group from the keto acid, and that produces ammonia. Then finally, the ammonia, which is toxic and can be harmful in the body if it builds up, is converted to urea, which is a harmless byproduct. What's left of the amino acid, because remember, you removed the amine group to form ammonia, but what's left of it becomes acetyl coenzyme A. And then remember that acetyl coenzyme A can enter the Krebs cycle. That's the molecule that does enter the Krebs cycle. And it can be broken down to make ATP. But if ATP is not needed by the body at that time, acetyl-CoA can go through reverse reactions to make either glucose or triglycerides. And either one of those two things can be stored for use later. They can be stored and then just used whenever your body needs energy at a later time. So that is protein catabolism. Protein anabolism is a process that you learned in Bio 120 in A&P 1. So we're not going to really get into it, but this is a process that's performed at the ribosomes, which are organelles that are found in all of your body cells. And it is a process that's directed by DNA and RNA. It is promoted by the hormones insulin and also HGH, human growth hormone. So your body is just going to follow the instructions that are in DNA and then RNA in order to build new proteins. And there are different types of amino acids. There are essential and non-essential amino acids. There's about 10 essential amino acids, and these are called essential because they must be present in the diet since the body cannot synthesize them in adequate amounts. And if you eat a complete protein, that is a protein that contains sufficient amounts of all 10 essential amino acids. And then there are 10 non-essential amino acids, which can be synthesized by the body in adequate amounts. These are formed by the process of transamination, which is when you transfer an amino group from an amino acid onto a different acid. And then that creates a new amino acid. As far as protein storage, proteins cannot be stored as they are. Oops, let me go back. They can't be stored as proteins or as amino acids. They need to be converted into a carbohydrate like glucose or glycogen, or a lipid like a triglyceride in order to be stored. Okay, so that's where I'm going to end things because that covers the metabolism of those three major nutrients: carbohydrates, lipids, and proteins.